Thanks for the introduction, Sel. Okay, so as just mentioned, I'm from a company called FizzerX. We're a local company here in Seattle. And the title of my presentation today will be Targeted mRNA Delivery to the Liver for Intracellular Enzyme Replacement Therapy. So what we do, we deliver mRNA to the liver to produce an enzyme that is missing in the liver. So this is the outline of my presentation. So first, I'm going to give you some introduction about our delivery system. So I'm going to show you the delivery system component. I'm going to show you some in vivo luciferase activity in mice using our nanoparticle and an mRNA that's coded for the luciferase. I'm going to show you some repeat dosing data, because it's very important when you have a nanoparticle that you can repeat dose and keep your activity. And then I'm going to show you some very exciting data on our disease model. So we work on urea cycle disorder, and I'm going to show you some therapeutic activity we got in our liver orphan disease model. So at FizzerX, we developed a system that we call FazeRX hybrid mRNA technology. And this system it's a two-vial formulation. Okay, so there is two important components in the system. So first, we use a diblock polymer that is designed for endosomal escape. So this particular polymer is a diblock, and there is a GalNAc targeting ligand. So we have a ligand on our polymer that is specific to the hepatocyte. And we have a portion that is hydrophilic air that's helped for the solubility of the polymer. And the key active part of the polymer air is this hydrophobic block that's helped for endosomal escape. And what's very cool about those polymers is in aqueous solution, they self-assemble in a micelle that is, let's say, 15 to 25 nanometer. And it's targeted to the hepatocyte, and it helps for endosomal escape or delivery of the mRNA. And this mRNA, which is the second part of our formulation, one vial is the polymer, the second vial is the mRNA. This mRNA is encapsulated in a lipid nanoparticle. And this lipid nanoparticle are made with the nanoassembler. So we have a very robust lipid nanoparticle system. And the beauty of this is we can use an inert lipid nanoparticle. So, because one of the inconvenience of the lipid nanoparticle is their toxicity. But since we use the polymer to release the mRNA from the nanoparticle, we could use an inert nanoparticle that is not toxic. And this nanoparticle shown like very good mRNA protection in serum. We got up to 24-hour serum stability in vitro. And the last one, and it's specific to the liver. So this system shown higher protein production in the liver. Okay, so this is the way the system works. So basically, we have two vials. We have a vial of micelle and a vial of LNP. We mix them together. We inject animal. This gets into the endosome and release the mRNA, and then we can produce the therapeutic protein. This slide here shows the progression of our system in the past 3 years. When we started working with this, let's say formulation 1, we were only using the polymer to deliver the mRNA. And we were getting, this is using the luciferase mRNA, so we were looking at luciferase expression in the liver. We're getting around 1/10 to the 7 photon per second in the liver. And we were super excited. It was 3 years ago. The polymer was able to deliver mRNA. It was great. And then over the years, we did some tweak to the polymer, tweak to the mRNA, tweak to added the LNP around air, and optimize the polymer to deliver the mRNA via the LNP. And now we have 5-fold improvement in our mRNA delivery activity. So we went from 10 to the 7 now to 10 to the 11 photon per second. And the data to here at 10 to the 11 are very tight. So we have very good reproducibility. So in real life, this is the way it looked like. So here we have luciferase imaging in the liver. We can see the bioluminescence is specific to the liver because we use the GalNAc targeting ligand on the polymer. So only luciferase expression in the liver. And here we have immunofluorescence. And you can see that we have very homogeneous distribution in the hepatocyte. And what is very cool, too, about the luciferase system is the expression is very fast. The slide here said after an hour, but you can start seeing it after 30 minutes. And it lasts for a long time. So we get very rapid expression of the luciferase, and you get it live. So, and this slide here is ex vivo imaging. Basically, we image the mice, and then you wonder if there is some luciferase expression somewhere else. You see, like, because the spleen could be hidden behind the liver or something like this. So we took off the tissue, put them in a Petri dish, and re-image them. And you can see that our system is only expressing in the liver. There is nothing in the spleen or other tissue. And this is another great advantage of using the polymer to deliver to the liver. Because if you use LNP alone, some of the LNP will show up in the spleen. But with our system, we only express the protein in the liver. So this slide here is showing you our in vivo dose response from 0.1 milligram per kilo all the way to 10 milligram per kilo. So a 100-fold range here. And we got progressive luciferase expression. Our system, too, is very well tolerated, less than 3-fold increase in the ALT level. So, and the key here, it's a great dose response. A great advantage, too, of this system is the ability to repeat dose. So here we use our LNP polymer system. We dose the mice weekly. So every week they got a dose of polymer and a dose of mRNA at 0.5 milligram per kilo. And you can see that the expression is very stable for 12 weeks. So no decrease in expression over time. And this is in CD1 mice that are like immunocompetent. Okay, so now let's go to the key part of this presentation is the activity of this mRNA hybrid system in liver orphan disease model. So first here, I'm going to introduce you the disease we're working on. So we're working on urea cycle disorder that are metabolic disorder that are devastating for the patient. So basically, those disease is there is a single gene mutation, and the people cannot make an enzyme to degrade some amino acid. And in this case of the urea cycle disorder, this produces hyperammonemia. So there's a lot of ammonia in the serum, and then those patients have neurological crisis, coma, and they will die. So it's very toxic. Ammonia in the serum is very toxic. So, and there is no real cure. So the only way those patients can get by is either getting a liver transplant. That is a very drastic cure, if you want. Or they can use ammonia scavenger, but those ammonia scavenger drugs are palliative at the best. It doesn't cure the disease. So by replacing the missing enzyme, we hope to cure this tragic disease. So this is an orphan disease or a rare disease. So the incidence is 1 on 56,000. And the average long-term survival rate is about 65%. At birth, like the patients have like some very severe symptoms like vomiting, seizure, coma, and they can even die from those ammonia crises. And most of the patients by the age of 12, because some live a bit longer, because there is different severity of the disease. By the age of 12, they have hyperammonemia crisis. So if you can replace the missing enzyme, then those ammonia crises will not happen and the patient will live. So, as I said, the only cure is liver transplant, and this is the current drug that are on the market, and Ravicti is just palliative. It's not really curing the disease. So, so as I just mentioned, so this happens inside the cell, And you cannot make an enzyme in a bioreactor and inject it. It's not going to get inside the hepatocyte. So by using mRNA, you can produce the missing enzyme inside the cell and then cure the disease. So, and since it's a single gene mutation, so you just need to replace one enzyme. So, and we think we have good formulation for this since it's a chronic disease, so you need to be able to repeat dose, and we can repeat dose with our system. So one of the key factor of this, preclinical or clinical factor, like as I mentioned, is the ammonia serum level. So we use a mice model that's called OTC FPF ash mice. So those mice shown high plasma ammonia level, okay, compared to the normal mice here. So normal mice here. So we use those SPF ash that have high ammonia level, and we treated them with either control mRNA. So it's an mRNA that has a stop codon in it that doesn't code for the protein, or we use a therapeutic mRNA that's going to code for the OTC protein. And using our therapeutic mRNA here, we were able to significantly reduce the plasma ammonia level at day 14, 21, 28, and 35. And this is a key data for us because based on the KOL, if you can reduce the ammonia level, then it's approvable endpoint for the FDA. So you can get your drug approved if you can reduce the ammonia level in the serum. So this is what we've shown here. We also look at another biomarker called erotic acid. So you can see here that the buffer and the stop codon mRNA or the non-coding mRNA, there is high level of erotic acid. And then if you use the therapeutic mRNA here, you get reduction in erotic acid. But the key is survival. And this data here show that the buffer or the control mRNA, the mice are gone all dead at 25 days, let's say, and the treated mRNA here, we have 100% survival after 35 days. So this hybrid mRNA system can lead to 100% survival of the infected mice. So another great thing too of our formulation is it's not toxic. We're looking at ALT level with different mRNA. Therapeutic mRNA, luciferase mRNA, or EPO mRNA, there is no ALT level, and there is also no cytokine production. So we have a well-tolerated system. And in summary, so here I just shown that we have a very effective delivery system to the liver. We're not going to the spleen. We have a well-tolerated therapeutic window from 0.1 to 10 milligram per kilo. We can repeat dose, and we have therapeutic activity in our OTC model. We got normalization of the plasma ammonia, which is a probable endpoint for that disease for the FDA. And then I want to thank the team because I did not do all that work. This work was done by the chemistry department at FizzerX, led by Sean Monahan, and the biology department, led by Mary Preeve. We also have a poster this week at the CRS, poster 314, where we're going to give you more detail about the polymer structure and the system. And I will be there too if you guys have some questions. And the CEO of our company is Robert Overall, and our CSO is Mike Liston, and I'm happy to answer any of your questions. Thank you.
So we have time for a few questions.
So maybe I'll start. So I'm going to jump in here because we have a question from our colleagues participating through the live stream. So this is from Jihan, who's interested in learning more about the GalNAc as a targeting agent. So you were mentioning that it provides delivery to the cytoplasm. So basically, Jihan wants, maybe you can speak more about the both.
Maybe I misspoke. I wanted to say it provides delivery to the hepatocyte. And then the polymer, which is hydrophobic block, help for endosomal escape, leading to the release of the mRNA in the cytoplasm. So sorry, maybe I was not clear when I was talking.
I was probably unclear in answering the question.
Quick question about endosomal escape. Were you able to image it? So once you put the polymer in, are you able to image the escape in the cytosol? Do you have data to suggest that it is endosomal escape and not just higher uptake or some pH thing happening?
Okay, we don't have direct evidence of that we really have endosomal escape. We have indirect evidence because we use an inert LNP that is not active by itself. And we know that the reason why this LNP is not active is mainly due to endosomal escape. The way that the polymer is chemically designed to, there is a change in the conformation of the polymer at low pH. And this is designed in a way to disrupt the membrane. So it should happen in the endosome. But we don't have clearly immunofluorescent imaging or something to really show what's happened there.
Thank you. Very interesting talk. I want to ask two questions, actually. One is, I'm not sure what kind of endosomal escape polymer do you use. Is it like one of the already published one or a new polymer? What's the main advantage compared to a variety of polymers already used for that topic? And another question is that, why do you co-administer polymer micelles and liposomes instead of modified polymer on the liposome surface? Thank you.
Okay, these are good questions. Due to IP reason, I cannot give you all the detail in our dye-block polymer, but I can tell you it's a dye-block polymer that is pH sensitive. It's designed with, let's say, carboxylic acid or molecule similar that's going to change charge with the pH. If you change the charge, then you change the hydrophobicity of the molecule, and then it disrupts the endosomal membrane. And your second question, why we use a 2-vial formulation instead of a 1-vial formulation? It's a hard question to answer. Because, this polymer, it's been designed to endosomal escape. And it's kind of the evolution of our polymer system over time. At the beginning, we were, 3 years ago, we were attaching polycation charge on our polymer to deliver mRNA. And we were getting success, 10 to the 7, 10 to the 8 RLU. But the polymer by itself, or the micelle by themselves, were not good enough to protect the mRNA in the serum. So they were very good at endosomal escape, but couldn't protect mRNA. So then we came up with the idea that the best way to protect the mRNA was to put this mRNA in a LNP. So then we started to design LNP, but we knew that a lot of LNP are cytotoxic, so we designed our LNP to make them super safe, but if you make them safe or inert, they don't really work. So then we mix them with the polymer, and that's why we have this 2-vial formulation. So it's a bit the story of this product development. Does that answer your question? Thank you.
Pierrot, I have a question. Very nice talk. I'm here.
I see you.
So my question is sort of related to the earlier questions, and I was trying to understand your mechanism of uptake of the delivery system. So I was hoping if you could provide a little bit more insight into how it works. So the polymer is GalNAc conjugated, so it gets to the liver through a known mechanism for GalNAc. The lipid nanoparticle probably gets to the liver in a similar way that's known. And then they have to be in the endosome at the same time for your polymer to work. So how do you control the kinetics of delivery? Can you give us some insight on that? And then I have another question.
Yeah, this is a very good question, and this is what keeps us awake at night, how this system works. You see, is the two particles, the micelle and the LNP, get in the endosome at the same time? Is the one endosome vesicle as an LNP, another endosomal vesicle as the polymer, they fuse together in the cytoplasm? We don't really know. We're trying to understand that. We're working hard at understanding how that system works, but we haven't figured this out yet. But we know it's working and it's safe.
Interesting.
Thank you.
I was wondering if you can also comment on the storage stability of your delivery system.
Okay, so far we have a frozen product. So the polymer is frozen, the LNP are frozen, they're thawed before injection, mixed together. They have 24-hour stability when they're together. We don't see a drop in activity that you mix them immediately or wait 24 hours before injecting them. So far it's a frozen product, and we're planning the stability study to move this forward. Thank you.